More brachiopod time. Okay, let's go. Can you spot the brachiopods in the picture? Yeah, the glossy little brown things on here. There may also be little flat ones here. I am not sure, but the glossy ones definitely, for sure. Okay, let's talk brachiopod internal organs, anatomy. Like you've seen the shells, or you're gonna see the shells. Yeah, a lot of the shells. So, like, how does the animal fit the bones essentially? And here's the answer. So, up here in the posterior, and so we're looking at the brachial valve of the posterior, which is where the feeding arms are going to usually leave an imprint. They're kind of stuck to that in life in some forms. In other forms, they're kind of free floating. In some, like the rhynchonellates, they can actually be sort of inflated and extended a little bit out of the shell to give you more area that you can feed from potentially. It seems to be an adaptation. And that's one of the groups that's still around, whereas some of the others had kind of a mineralized support. So you know the whole thing, it was just like a solid piece. So you know the whole lophophore was not moving outside the shell. Yeah, it was just opening and closing. So down here at the posterior, you've got the mouth. There's the little mouth opening that I'm circling. And this is going to be equivalent to the stomach. There are sometimes additional digestive tracts that are going to be off here to the side. Next to this is kind of a parallel system. These are going to be the gonads. Those do move around, and sometimes those will stretch all the way down the side, we think, in some of the earlier versions. Those would then sort of have some kind of opening where they can feed out and get into water circulation because they need to sort of breathe those out. Up here at the back, you have some little organs that are doing the job of kidneys effectively. Nephridiopores. On some of these organisms, they will have an actual, this there it says right there, a brood pouch, which is a place where the little fertilized embryos can go through their earliest metamorphoses and yeah, start to grow. Here's our little, I can't remember if that's a disconid or a lingulid, but it's adorable. Look at it floating around there, moving its shell like that because it's not, it doesn't have teeth along its hinge, so the two plates can slide relative to each other. All right, and then on top of all this, this is the big feeding arm, and so it's going to come out around there and then go back up to about here. And sometimes there will be structures in the center, which I believe will serve. So it's going to be pulling in water from the center, and then it gets flicked to both sides. So those help sort of divert the current, deflect the current, and they would also add some turbulence to it. Maybe change up the speed of the incoming flow, and that could make filter feeding a little more efficient, would be the hypothesis. It needs to be tested formally. All right, so here is a fossil, and see if you can put the organs on there. So you can see, boop, boop, boop. There's our, that is our lophophore. So that is our feeding tentacles. Great. Where would the mouth have been? Probably right about there, maybe right about there. And so then the rest of this is going to be the body. Were there digestive glands spread out here? Maybe, but probably only to about here. It looks like there are little passages where those could sort of squeeze between. Where were the gonads located? Possibly up in this area or right around there. Yeah, that would be about right if we're comparing to this particular group of modern brachiopods. But yeah, there hasn't been a good reconstruction with all the soft parts in a brachiopod related to these. So there's not one where you can see for sure where the gonads are. And there's so many different kinds of brachiopods, they could actually move around a bit in the anatomy. Yeah, it's hard to be certain of that. So this is what a brood pouch looks like. So the mouth is there, and there's going to be these little tentacles connected up to it. And so at the end of those, you're going to have a bunch of little embryos that sort of anchor down with their little, remember, it's that little lobe that's going to become the pedicle. And there's a little three-lobed structure, and then they are going to develop into little brachiopods in there. just the earliest stages. So by the time they come out of the brood pouch, they're going to at least have a little bit of a protegulum, which is that shell, like you see up there. Only like the original version is really small, and it's just like a little cover for the soft parts that are here. So maybe about that size. Yeah. All right. But what's going on under the hood? Well, a lot of things, right? So this is a map of the nerves in a modern brachiopod. And so there's something that is kind of like the equivalent of a higher brain, if you like, I would say, up here. And then you've got this ring brain down at the back, and that's going to circle around the esophagus. And that is going to be a very familiar arrangement if you've looked at a lot of anatomy of invertebrates, very similar to what you'd find in things like mollusks, for example, which are probably fairly close relatives. And then you've got sort of branching nerves that are going out, and they're going to be nerves that are going into the parts of the lophophore that are going to enable the tentacles to move and catch food and do the things they need to do. Looking at the lophophore, so we have to imagine it, but we can see its imprint on some of these brachiopods, or we can see like a spiral kind of shell material apparatus that the lophophore would have been attached to in life. So we don't have to completely imagine it out of nothing in every case. It's one of the better marked features that's no longer there. And then at the center of that and at the back, that's where the mouth would have been. Yeah. The tentacles on here work more like the tube feet of an echinoderm, in that there's a little pouch at the bottom, and if you squeeze this pouch, then it's going to inflate the tentacle with water, and so then it can stick out, and that would then make all the little cilia beat in time, and you'd be filter feeding. So looking at the imprints on the inside of these, there are a lot of different ways to support the lophophore, and so all the stuff that is the same about these is generally just the outer shell, the thing that's covering over the precious lophophore. The actual animal in every case is usually confined to about the back third or quarter of the shell. These are all the living parts. It's just got two arms that stick out to here. There is a thin sort of skin-like mantle that's actually going to cover the entire inside of both valves. And that's going to be the thing that is going to build up more shells. We're going to build it up from the inside, and we're going to build it up at the edge of what we've already got there by sort of poking our little mantle out just barely, and we're going to lay down some of this material underneath the mantle. Because if you think about it, these things are living in the water, and so if they get a calcium carbonate and, hold on to it, if they just sort of set it down, it would just float away. And then what? Then you wouldn't build a shell. And so what they do is they trap it underneath kind of a layer of cells, a watertight layer of cells. And so they just stack all these up in there. And then they cover them over with a layer of protein, or make sure they're really well attached to each other and just don't ever get completely exposed to the water. Because if they do, then they can potentially dissolve. Yeah. But yeah, you've got some of these loops that go back and around and up together. That's a really neat one you find in the pterobrachilids. These are thesidiids, and they got this crazy looping around here and around there and up to the beginning. That's absolutely nuts. You've got crura on some of these early forms, which are just there to hold up the start of the lophophore, which then is not supported the rest of it. And in some of these, like spirifers and spiriferinids and athyrodids and atropids, you're going to have a little spiral support, and there's a couple of pieces that connect this up to the back of one of the valves. And so this is going to support the lophophore and enable like a lot of lophophore to do a lot of filtering in there. So this may have been a sort of a maximum extraction of resources kind of modification, if we're trying to think in terms of what was evolution trying to do here. When of course the answer is evolution tries to do nothing, understands nothing, and if it works, it lives, and if it doesn't work, hey, whatever. No big deal. And then when you're looking at the imprints of the feeding arms of the lophophore, um, got to remember that those would have had these little side tentacles coming off. And the side tentacles are usually about long enough to touch or just barely go outside the outer margin of the shell. And then, yeah, when it's in sort of filtering position, it may be a little more up like this, something like that. But yeah, don't. So these are the feeding arms, but don't forget the little tentacles on there. So if it seems like everything is crowded toward the center of the shell, it's just because you're not taking into account the tentacles that are out there, because those would have filled it out a lot more. How do these little tentacles work? Well, you've got a bunch of little beating cilia, and you got sort of a triangular cross section here with long ones on the corners. And these are going to be beating in time, and they're going to beat in such a way, and also on the sort of mantle, if you like, there's going to be other little cilia, and so those are also going to be helping to pull water in. So as long as those are all moving in a coordinated manner, that will pull water in, we think, from the center of the shell. That's how it works in the modern ones. And then push it out the sides at the sort of posterior and sides of the shell. Yeah. All right. What about this brachiopod shell I've been hearing so much about? We might as well cover like how it's built, because it's built in kind of a weird way compared to how a lot of other stuff works. Just like, you'd look at stereom and think that's normal if you only knew about echinoderms. And then if you look at anything else, you're like, wow, stereom's really weird. It's full of holes. Can you spot the brachiopods here? They're little craniids. Some of them are actually open. They've lost their top shell and they're presumably dead. Those are the cemented dorsal valves, but the rest of these seem to have their ventral valves still on. All these little sort of orangey lumps on the rock, and those are all little brachiopods. Doing very well, thank you. Okay, so when we're talking about the shell, we've got a couple of terms that tend to come up in the literature, and these are here so that if I slip and say something like this, or if we are looking at these brachiopods up under the microscope, you have words to describe the things that you are seeing, because that's all these terms are. They're just descriptors. So the very first formed shell is going to be called the protegulum. So that's PR, that's up here. And then after that, we've got a set of generalized words that apply to metamorphic creatures with some kind of shell. So these words are used to describe coral metamorphosis and brachiopod metamorphosis, not as often for mollusk metamorphosis, although occasionally, yes, they are, and other creatures as well. Yeah, generalized useful things. So you got your protegulum here. That's the earliest form. That's going to protect little baby brachiopod, or at least protect its vital organs. The brephic shell is going to be the next thing that's made. And so it's got to be brief because it's still really young. Yeah. And you will often notice a different texture in the brephic shell. In fact, that's the only way to notice it. There's not like a hard and fast guideline, like when it's 1 millimeter big or it does a particular thing, you can say that's the end of the brephic stage. These are squishy. These are pretty squishy terms. But when you do see visible differences in the shell, these are the terms you can use to describe the different zones. And it's just like describing a target for darts. Protegulum is the bullseye, brephic shell is the ring around the bullseye. The neanic stage is the next one. That's like your juvenile stages. So that's just the next ring out. And then the adult, or you can call it ephebic, but that's a horrible word when adult is right there, is going to be your pattern of growth that continues on for the rest of the shell. So all these changes from protegulum to brephic to neanic to ephebic or adult, those are going to happen in like the posterior less than a millimeter of these shells. And often, if you look at those under the microscope, their structure is really different than it is on the rest of the shell. And this is important in classifying brachiopods because they tend to follow. It's kind of hard to change your early development and easy to add another tweak to later development. If you want to think of how genes work in those terms, as if they're little day laborers or something. And then with some shells, they'll go on in adult form for a while, and then just before they die, they'll start doing other weird stuff. Like they'll get really thick or they'll get really curved. And there are some body parts that don't appear until late, and so we call those gerontic, old age development. And so there you go. You got developmental stages or names that we can apply to different parts of the shell that developed during those stages. Since we don't have the soft organism, we kind of have to, base it on what we can see. And for some brachiopods, you will not be able to tell clearly where one of these ends and another begins. There are some that may only have either a brephic or neanic, and you can't tell the difference, in which case, I guess you just say it's brephic and they have no real neanic stage, or where the neanic stage isn't really marked. Yeah, stuff like that happens. There isn't a better system, but it would kind of be nice if there was a more objective system that you could use that would always work. Yeah. So sorry about that. I don't think there's one that can be applied to everything and will still work, just because preservation is really different and it's based on what the shell looks like. And if you could just watch these things develop and see all the soft parts developing at the same time, you could probably tell a lot better and make a lot better judgment on this. Okay, the brachiopod shell, when you see one and it's a fossil, like a lot of times if you're trying to get it off of a piece of rock or something, it'll just shatter because they can essentially you can't bend them very much. This is like depth that you can push a probe into it without it breaking, and the depth is not very far because you can deform them a little tiny bit and then they just shatter, they just fail. And so this is what the dry shell does. But these things are never dry in life. They actually have a lot of water inside the shell. And the effect of that, at least in some brachiopods, some of them were really thick and almost certainly really rigid and kind of like the top graph there. Like the insides were probably bone dry. But in a lot of the ones that you would see now, like lingulids, the shell is actually pretty bendy when it is still wet. And if you dry them out, then it becomes much more brittle. So they can take a wide range of horrible bending and just no big deal. They just bounce right back. Yeah. All right. So the shell is going to be built up, like we said, underneath this mantle layer. And so you're going to have these mantle cells, and they're going to be transporting minerals. You'll have the basal lamina being there. It's like a little attachment surface for them. And these are like a little epithelial kind of layer, but they're epithelial on the side with the shell, so they're almost, yeah, they're not endothelial, but they're on the inside epithelial. And they are going to build up the shell in layers. So on the outside, you're going to have a periostracum, which is sometimes going to have like a protein layer on it, and that's going to protein, you can make proteins that are completely insoluble and are much more durable than any shell. And often proteins will survive better than things like shells and coral skeletons. The proteins can be really resistant in life and after death. So periostracum on the outside is going to be there. Then you're going to have a primary layer. Typically, for most of the sort of calcitic brachiopods, that is thought to be aragonite, if I remember right, which is like the long, sort of spiky form of calcite. And then we're going to build up these sort of fibrous layers. Each cell is going to secrete more of this. So it's going to just make a mound of shell material. And then as the mantle grows, this cell is going to move over here and it's going to start building up in this area next. And so they're going to thicken up the shell. And so this is how they'll work. They'll thicken from the inside and they will thicken and elongate at the edge. And that's how they're going to build up brand new shell. Yeah. So looking at our shell, there's our brephic, our protegulum would be in the center or the bottom of this brephic shell. You can call this like a neanic shell, I would say. They're calling it brephic here. And then adult shell. Yeah, that's fine. But you can actually, if you take a cross section, so this is like you got a brachiopod that's like this, and you slice it right down the middle. And so we're going to turn it so you can see what's going on up at the nose. The first formed shell, this is what I would call the protegulum. I wouldn't call this whole great big thing the protegulum. I'd call that brephic. So there's your protegulum, and then these have another little shell zone right here. That's the next bit of shell that they're going to build up. Here's the first bit of shell on the other valve. And they're going to build up the teeth in layers. They're going to build up the shell in layers, and they'll just keep adding to those throughout life. And so they get bigger by extending the margins and thickening the shell, which also makes them stronger, I guess. So that's not so bad. But the mineralogy of these things is kind of interesting because they are doing biomineralization, but they do it in a weird way. So if you look at some of these things like linguliforms, like discinids, brachiopods, if you look right at the first formed shell, it's going to be covered in like a mosaic, these little flat sort of pieces, little tablets they call them. And these tablets are made of silicon dioxide. So even though the shell of a discinid is going to be calcium phosphate when it grows up, it's got a whole bunch of little, it starts out with a bunch of little kind of loosely joined plates of glass, silicon dioxide. Literally starts out in a glass house. Isn't that neat? And then it abandons this completely for the rest of its life. It could be making a silicon dioxide shell. There are animals that do that. But no, it doesn't do that. Potentially because there's a little more availability of calcium carbonate in the water. Because as long as there's CO2 in the air and as long as there's calcium in runoff, which there always is, you can always make calcium carbonate. Then you're going to move toward these little smaller tablets, and these may not be silica in the brephic shell. These may start to become something like calcite. And then the stuff around them is going to have little tiny grains that are going to be calcium phosphate even. Yeah, just an insane way to do things. And then there's going to be real strong growth lines because like each of these it's going to grow and then it's going to pause like when insects are doing metamorphosis. It's going to grow to a certain size and then it's going to think about what it's done, rearrange what genes need to be expressed, and grow the next size. We don't get to see what happens to the animal. There's probably at least some change to the soft tissue inside at each of these points. We just can read the lines out like on a dead tree stump. It's not as interesting as watching the entire tree grow, but it tells you something. All right. So these are, this is a modern one. You can see the little tablets coming off, the little silica tablets. And then here you can see some older fossil shells. And they don't have the tablets always in place, but like, look, when the tablet comes off, it leaves a little rim around where it was. And you do see a whole lot of rims that are the shape of some of these tablets. Interesting. And so if you're looking at the absolute protegulum, here we go. You can see the little holes where some of these little possibly silica tablets were. We're not sure and how Paterula started. And these are things that would change on diagenesis potentially. And then look at that. The edge of the first form shell, you're going from circular little tablets to little half-moon shaped tablets. Look at that. To little rhombus shaped tablets on the rest of the shell. Yeah. So it is changing potentially the mineralogy, like what minerals it's using, and it's definitely changing the way it is organizing these things, expressing different genes to do all of this stuff, just as it's growing the first couple of millimeters. And that is deeply weird. So inside this system, to make a hard shell, you've actually got the remains of several different overlapping systems to make a hard shell. And it's going to turn them on and turn them off at different points in its life. So it's not just one way to do it, it's a bunch of ways. Which means there were probably things before this that were maybe just doing one of these. And if we had better fossils from the early Cambrian, like 540, 530, 520, if we had body fossils of things like brachiopods that were that old, if they even existed, they might have the keys to this. What also may be happening, though, is that you may have had something like a protein structure. And the protein structure is going to change as it gets bigger. You're basically making small circular plates, medium-sized moon plates, and larger rhombic plates as you get bigger. That just may be an efficient way to grow. And it may just be that learning how to attract minerals to those plates as they're being assembled, that may be a thing that was learned later. So it may have just been little chitin plates or something like that in essentially a chitin shell, like an arthropod, originally, that then figured out mineralization. Very interesting to actually study this sort of stuff. But there's not that many places in the world with really, really high quality Cambrian fossils, which is the thing that you would need. Now you look in a recent one or a later one, and they're going to look like this. Once you're inside, they have these great big sort of fiber looking columns. These are little fibers stretching up. Here's our little columns where they extend along. And so we're going to switch. We're going to do fiber growth for a while, then we're going to do column growth for a while, then we'll do fiber growth again for a while, and then we'll do column growth again for a while. We'll just alternate back and forth. So every time it's growing, think of it as like a growth cycle. And it'll do column, 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 and then fiber, 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 fiber to spread out, and then column, column, column to get thicker, and then fiber, fiber, fiber to spread out, and then columns to thicken up. So maybe the fibers are the thing you do in the spring, and the columns are the thing you do in the, winter and autumn and the rest of the year. Yeah, it would certainly make some sense. But if you look at the earliest ones, brachiopod shells, they don't quite look like that. So these are two really early, kind of problematic shells, and don't stop, never stop. Yeah. They have little, this thing, it's like a column structure. And sometimes these columns are hollow, but sometimes there are also hollow tubes in between the columns. So these look more like what you would get in an echinoderm shell. These may just be an artifact of how the shell was put together, and it may be that the stuff that's in between these columns was easier to dissolve, and so this is the part that's left. But it's a peculiar skeleton that you find in some of the earliest true brachiopods and some of the earliest things that are probably kind of sort of brachiopod-like, but maybe not precisely belonging with brachiopods. They're in the gray area, the crown group, if you like. But yeah, that's an early shell shape. So we got these are our sort of fibers spreading out, and these are our columns stacking up, essentially. And if you want to look at lingula, so this is a lingulid that's up here. Lingula is going to make this sort of network over here, and the network's going to be, I think, mainly protein. That's these glycosaminoglycan proteins they're talking about here. And these little lumps in here are the little lumps of calcium phosphate. So even though which is the other name for that is apatite, right there. So even though it looks like a nice smooth shell that's made out of calcium phosphate, it's just this crazy net with a bunch of little tiny balls of calcium phosphate that are kind of glued together and stuck to the net. When you get really close to it, it's a very strange organization. So yeah, brachiopod shells, they just look like a smooth shell if you don't look very, very close or very hard, but look closer and they are really wonderful. So here is what you see on some of the earliest ones, these acrotretids. You see a bunch of different shape and size little imprints. So you got these little hemispherical imprints, and so they're thinking, well, based on what we see now, the most likely explanation is there were a bunch of possibly calcite spheroids, because calcite will pack in a little sphere. Asymmetric calcite loves to. So maybe these are little calcite, like little pool balls that are then sort of glued together. And that's the main construction. There are also these long sort of vesicles, and so they're speculating that these may be composed of protein polymers with little sort of tablets, itty-bitty tablets of apatite, which there's some you can see around here, on the outside of it. So this may be the sort of early version of the adult skeleton. And then you'll have little calcite covered discs over here. You'll have proteins like mucin that are appearing over here. And you can find bits of these on several different existing lineages. And when you're going back and trying to look at a very early shell whose chemistry may have been altered by diagenesis, like the way fossils change, it's potentially tough, potentially kind of difficult to tell what's going on there. But yeah, looks like the process of building a brachiopod shell was really just difficult, and it's bringing a lot of different things together and throwing them all at this problem, and they're all helping a little bit, but yeah, it's got a lot of different ways to solve this. So even though it seems like silica bones and phosphate bones and calcium carbonate bones are all really different ways of doing things, you get some organisms like brachiopods where they have elements of all of these things happening at different developmental stages. So yeah, neat little microcosm of what can happen. Now, if you're looking in brachiopods, and let's jump all the way back up here to the picture of the interior. There we go. You'll often see, so this is the area where the muscles would have attached. Underneath the muscles, you get this sort of wrinkly looking shell buildup. And often when you're looking at the shells, if you've got the inside of it, you'll see that that's a different color. It's actually got a different structure to it. So these are, so the muscle fibers are on top. There's actually a little layer underneath the muscle fibers. And this is connective tissue. It's acting a bit like a ligament, but also there are going to be cells in there that are building up very, very thin calcite. So weirdly, this muscle attachment scar is going to build up over time, even though the muscle never like leaves so it can be built up. It's building up under the muscle while the muscle is still attached, which is kind of tricky when you think about it. And so the pattern of muscle growth is sometimes diagnostic for brachiopods. Like they'll have a certain way that their muscle, these little lines will sort of branch in there. Yeah. All right. And like I said, they leave different colored shells. So the places where the muscles attach are all marked in colors here and colors on here. And see that? See how the shell is all white there? This is like a section through the shell. And see, look how white it is there. The shell material, it's still mostly calcite, but there are probably different proteins in there, and there are certainly thinner, more compact layers that are forming underneath the muscles because they're like under a lot of stress all the time. And yeah, it's different sort of cells doing a similar sort of thing in there. Or maybe you can say that the opposite way, and it makes even more sense. But yeah, you can find the muscle scars, especially if you section. It's pretty easy to find. Yeah. All right. And the muscles will attach like this. You'll have the ones that pull the shell shut are going to be attached to both of the valves. And so they're just going to tense up, and that's going to close the shell. The one that opens the shell, that's kind of counterintuitive. So we're going to use these guys as like a fulcrum. So that's just going to be standing there. And then we're going to pull down on this side. We'll pull down like where my fingers are in order to lift the outside. So we're going to pull down. Our muscle that opens has a giant connection right in the middle of the ventral valve, and a little tiny connection just on the tip of the dorsal valve. It actually wraps around this little thing that's called the cardinal process. And these little grooves there. It's kind of like a tendon, a muscle attachment point. It's not exactly a tendon because these aren't people, but it's the same idea. So yeah, we're gonna pull down this. It's kind of like the way a step garbage can. You sort of step on it. There's a little thing at the back that's going to pull down. That's going to lift the lid up. That's the idea. For brachiopod shells. Also, on brachiopod shells, some of them will have these hinge spines, or you can call them cardinal spines. And sometimes they have valve spines. Now, usually the valve spines, so those vary. A lot of those are closed off from the inside. So they were like a spine for a while, or they may have a spine thing in there and they may be loosely fed from the inside. But, eventually the spine dies and it's no big deal. But the spines along the hinge are hollow. And so there's actually, we think, some of the mantle that's filling up these spines is just like armored little fingers of mantle that reach out. And sometimes these things will, like, look at these spines. They're sort of wrapping onto and holding, kind of gluing the brachiopod down to a substrate that it doesn't want to leave. Like here they are as well, reaching out and grabbing. So even if the brachiopod's not attached, it'll still grow these spines. And just think of them as like little sticky tipped fingers. But if they encounter something that's hard, they will then cement themselves to that. And potentially that keeps the brachiopod in place. And if it's in a good place and it's made it this far in life, feeding in that place, then stick with what you know, brachiopods. Yeah, never change. Yeah. Okay. Where do baby brachiopods come from? Well, we talked about the brood pouch that you find in some modern ones. The brood pouch doesn't leave an impression on the shell that we can discern on the modern ones. So there's no good way yet to be sure of whether this existed before, like last year, or if this was something old. But you do get on a lot of older ones, orchids, maybe this is a terebratulid, yeah, these sort of gonad lattices. And so this is going to be where all the little egg and sperm generating cells live. And then they've got a series of tunnels. And those things are going to pass out into the water column and be released when it's the right time, when love is in the air. And then just to remind you that water is going to come in from the center, we think, based on the modern ones. And it's going to go out at the sides. Could some older ones have done it a different way? Potentially, but I think they make more sense if you assume this is conserved all the way through. Yeah. Okay. So these are going to be for an activity that we do in class. I'm going to give you some brachiopod interiors, and we're going to try to make a little description. But this is just showing you that each of the different parts has a name. And so you can say, all right, what's happening on the wing area? And we can say, okay, the wing is from here to here to here. And so, the wing has a bunch of little bumps on it. And so, yeah, that's exactly what you'll say. And so normally when you're describing these shells, you'll move from posterior toward the anterior, and you'll move from the center out to the edges. And you'll just describe each part. You'll be like, does it have a nose that sticks up beyond the interarea? In this case, yes. And then good, nose projects beyond interarea. Yeah, that's part one of the description. And so you're just going down a list. It's like a checklist of these parts. And you just say, does it have this part? Is there something weird about this part? And then you have to kind of say what's weird about it, if it is weird. And yeah, so like... look at the median septum. Can you find that? This is the ventral median septum. And so ventral median septum would be right here. So it's connected to this little sort of flat bit up here. That's the apical callosity, which is like the brachial valve's like a ball and socket joint. It's going to turn in this. And so the apical callosity is like the socket, and the brachial valve will turn up like that. So it's got a little bit of muscle reaching up in there to sort of pull it down and lift up the valve. Yeah. But you can see this one has a very long median septum. Look at that. This one has a fairly long one, though it starts to get broken up as you get toward the end. This one, it has one, but it's tucked up in shadow under the nose here. This one is also super tiny. It stops right about there. And here's another one that's super tiny. So those are features that you can use to actually separate different groups of brachiopods. Do they have this feature? Is this feature like longer than half the shell, or all the way to the end of the shell, or really short, like a quarter of the shell? Those are the ways that it is done. The ventral valve, not as useful for taxonomy, deciding who's what and how are they related. Dorsal valve has the imprints of a bunch of organs in ways that we don't completely understand. And it has the feeding arms. And it turns out the feeding arms were really different on different groups of brachiopods. And so we can group these and we'll put them in a different genus if it's like you can say different words about them. If it's just like bigger or smaller, we would say that's just like a difference in degree and we would normally keep them in the same genus. But like look at the median septum. Here it's super short, but it ends with two little, they're not testicles, but they're two little lumps, yeah, of shell at the end of it. That's weird. It's the only one that does that. Look at this one. It goes up like a ramp, and then the ramp is jagged at the end. And you can look at a bunch of these, and they all do that. So that's like their thing. It's not broken off. Insane. Like, okay, but that's very different from this. Yeah. And then you got this one that's super low to the ground, and then right at the end, the anterior, it pops up into this giant, like, Tower of Babel looking spike. Yeah. And then this guy, it's curved a little bit off to the left, and it's kind of short and low and smooth. Actually, pretty close to this one, but it doesn't have those two large nubs of shell at the end of it. So that's one feature and how it's different. This is a map for some of the other features, and that's what we're going to be doing in the practical, is trying to categorize these things. Because since we don't have genetics for things this old, this is the only way you can figure out who's related to who, and how did all these things get here? Yeah. So here you go. This is the updated version. You'll be working from this. So this is the shapes they are outside. These are all the parts that they have on the inside. And these have more parts than the ones on the previous slides because they're more advanced, better, just better. Yeah. Yeah, and there's some study questions for the end. So, all right. Hope that helped. Brachiopods. We've got one more section on brachiopods left.